The first speaker is the president of EPFL, the Swiss Institute of Technology. That's a leading school when it comes to life sciences and neurosciences. So it's a pleasure to have as the opening speaker of the day, the president of EPFL, Professor Patrick Aebischer. Welcome. Thank
you. As I say, I think neuroscience is a fascinating science subject. I would say it's the last science frontier. And today we're living a very interesting time in brain science in the sense that multidisciplinary fields amenable to interface with engineering and basic science are helping us to finally understand the most fascinating machine, the human brain. But also it was said, with the aging population, we need new and more efficient treatments for diseases like Alzheimer's, Parkinson, epilepsy, and very many others. The challenge is, when you look at the last maybe couple of decades, we've made tremendous progress in the way we tackle cardiovascular disease, cancer, but we have to realize we haven't made the same progress for the treatment of neurological disease. Just for Europe, close to 100 million, 80 million Europeans suffer for brain-related disorders, being neurological or psychiatric. Just the cost for Europe is estimated at 800 billion euros per year. For Switzerland, a very small country, close to 15 billion a year. So we have a major economical challenge with the aging population and the rise of neurological disease. What have you done? Science has progressed. This is just a graph that looks at the number of publications in the field of neuroscience. So you could see that from 2000 more or less, this has increased quite significantly. And this is good news because you need great basic science to try to tackle those issues. However, if you look at the number of FDA-approved drugs for neurological disease, this slide is a bit depressing. This is the number of new drugs that have been approved by the Food and Drug Administration. Same thing for the European community. You could see here that with the exception maybe for migraine and pain and analgesia, which is often reformulation, we have not found really new innovative drugs, maybe with the exception of multiple sclerosis for the last 20 years. So this is an issue for mankind. Now you look at this other graph, which I think is also very telling. In the 1950s, $1 billion investment would get you 50 to 60 new drugs. Today, you have to put more than $1 billion to find a new drug. And the drugs in the nervous system are probably the most expensive one. So we have to find a way to better design, faster, and to some extent, cheaper. This is another a bit depressing slide. You can see here the number of pharma industry that have left or closed down, shut down their basic neuroscience department. Look at AstraZeneca, Novartis, Merck Serono, and again, showing that we need to come with a new way to tackle those problems. But there is one good news. For example, if you look at the treatment of Parkinson's disease, essential tremor, one man, Dr. Alim Benabid, who will be the next speaker, has given us hope. Through his really innovative approach of deep brain stimulation, I mean stimulating very specific areas of the brain with electrodes like in a pacemaker but in the brain, he has shown major positive outcome for essential tremor, for Parkinson's, for dystonia, but it shows that even it could go beyond. Depression is a potential target for deep brain stimulation. Some people call it electroceuticals. So I think it just shows that it's not only drug, but we need to take a broader approach for the treatment of neurological diseases. And that's what we've done in Lausanne at EPFL. We've launched this initiative called Neuropolis. The idea is not only to do the great neuroscience, but also to put emphasis in computer science at the interface of neuroscience. We call neurosimulation or neurotranslation. Let me develop some of those. Just when I got out of medical school, when we wanted to simulate Parkinson's disease in a rat or an animal, we had to use neurotoxins like 6-hydroxydopamine or MPTP. Those create symptoms, but they're not really the cause of the disease. What has changed? If you look at from 1997, that was the first gene discovered related to Parkinson's disease. Today, we have 5 major genes that are related to Parkinson's disease. Let me just do one very simple example on genetic risk factors in sporadic Parkinson. You can see here 2 genes, alpha-synuclein and LRRK2. LRRK2 is interesting for this part of the world because the prevalence of Parkinson's disease related to LRRK2 mutation is high in the Middle East. But it is potentially a target for pharma because it is a kinase and a GTPase. So this is early days, but it is thinkable that we can modify the way this disease will progress by affecting, modifying, for example, LRRK2 in people that suffer from LRRK2 mutations. But we have now tools that we didn't have before, and those tools are high-performance computing tools. Let me give you one example that started in my school, and it was really the idea of one of our professors called Henry Markram that had this brilliant idea to try to use the power of supercomputing to do what we would call reverse engineering. Can we build a model of the brain? Can we simulate, by extracting the data of neuroscience that were gathered by scientists, the way a brain functions? And for this, you need a very big computer, specific computer. But today, without going into the detail, he has the capability to produce simulation of up to 250,000 neurons with several hundreds million synapses. Now, the brain contains billions of neurons, so probably you will need a bigger computer to tackle this. But let me give you just one example, but also to say that this was sufficiently interesting that the European community has decided to fund this project to the level of €1 billion for the next 10 years. This is a consortium of 242 researchers in 134 institutions in 24 countries that are working together to try to understand, by simulation approaches, how the brain functions. And this is one example. This is what we call a cortical column. This is kind of the CPU of the brain. This is when you put all the data inside, and you could see this neuronal activity, electrical activity. Now the idea is that you come here with one cell, one receptor. You try to modify it, and you try to see how the cortical column functions. This is a potential tool to develop new therapies for pharmaceutical industry. We are in the very early days, but just to say that this is something very new happening to the field of neuroscience, where computer science, mathematical modeling, and neuroscience are trying to put together to try to simulate the way our brain functions. But let me just emphasize the last part of my talk to what I call the needed effort for neurotranslation. So at EPFL, we decided to concentrate on 3 aspects: neural prosthesis, gene therapy for CNS, for central nervous system disease, and what we call nutrition-based disease prevention. Let me develop the first one, neural prosthesis. When you look at what the medicine has brought over the last, again, 20 or 30 years, it's quite remarkable. What we call the bionic man. So if you take one example, the sensory prosthesis. Cochlear implants have made tremendous progress in the last decades. There are today more than 200 people that benefit from those devices. But with the progress of microtechnology, electrical engineering, and so on, those processes could be smaller, less invasive, and you can see that this will lead to, I think, very significant solution to hearing loss. And again, helping the aging population. Retinal implants are in their early days, but it is conceivable that for disease like macular degeneration, those prostheses could replace partially vision. So this is a very exciting field. You will hear from Professor Miguel Nicolelis, one of the world experts, that we can think today of brain-machine interface, like having robot artificial arms connected to the nervous system and controlling your environment by reading what your brain wants to do. This is a very exciting field, but that shows, again, that brings very different technologies from engineering, electrical engineering, microtechnology, material science, computer science. So you will need this kind of concentrated effort to develop those. This is one example, one of our professors that has developed a wheelchair that is controlled by the brain. So this is an EEG, and you can see that this individual can now start to think about a movement, so that he's now thinking about going right. This is detected by the system and controlled the wheelchair. So those are the kind of brain-machine interface that you could start to think that could help a paraplegic, that could, or tetraplegic patient in this case, that could really control his wheelchair by the power of his mind. One other way is one of our professors, Professor Grégoire Courtine, is working on spinal cord regeneration. This is at the rat level. This is a rat that was sectioned and was paralyzed in his posterior limbs. And you could see that by combining electrical, chemical stimulation, but also with this robots, a rehabilitation, you could think of helping. And he's now, in fact, preparing a clinical trial in a paraplegic patient. Those were unthinkable a couple of years ago, but this is again an example where you put biology, electrical engineering, neurochemistry, rehabilitation together. So I think it is really this holistic approach that can bring things. Another example is gene therapy. Gene therapy today is finally a clinical reality. This is the number of clinical trials in the world. You can see the majority are for cancer. Neurology is very small. So now, I don't have the time, but we have been able to cure mice that suffer. We went now into primates. We can see that we can infect more than 50% of the primate brain, motor neurons. So it is a way to show that we can now think about going into the nervous system of people with gene therapy. Let me give you 3 targets that we are now currently working at EPFL, and the way we hope that we will be in human in the next 5 years. Motor neuron disease, I've said for ALS, silencing this mutation of this gene called superoxide dismutase, but SMA, spinal muscular atrophy, a very difficult disease, prevalent in this part of the world too, where we can express a gene that has been mutated and lost its function called SMN. Well, there's another disease, we will not go again into the detail of what we call amaurosis in the eye. Huntington disease is also potentially amenable for gene therapy. So I think we will see in the next 5 to 10 years gene therapy going into human. And the last way that I'm quite interested, again, where I see a future for this part of the world, is nutrition-based disease. Now you could say, how could you do nutrition? Let me give you an example. There was a couple of years ago an interesting paper that was published in Science showing that a molecule called Resveratrol that you find in the skin of red grapes that has very significant effect, for example, in weight control. We were quite fascinated to try to understand how this molecules affected a certain gene pathway. But it's interesting, this is a little worm that we call C. elegans, very much used in laboratories. You can see that by giving this molecule, we can extend the life expectancy of those worms by more than 40%. This is very intriguing when you think about the context of gene of aging disease. Because that's where you could start to think that if you could increase, and maybe there are molecules there that can affect the overall metabolism, and we believe that, for example, mitochondria have a key role in this aging process. And we know that neurodegenerative diseases, for example, are related to mitochondrial dysfunction, at least some of them. So we've tested this compound in Alzheimer mouse. And again, without going into the detail, we've shown there is a significant improvement in memory loss in Alzheimer mouse disease. So in summary, what I would like to say is that we need to show clinical impact. And those will come with a combination of great neuroscience, I would say the classical neuroscience, getting computer science, simulation, neurotranslation, be it gene therapy, neuroprosthesis, nutrition-based prevention. So I think we have to see it a much more holistic way to tackle those problems. And that's what we want to do at EPFL. So how? First and most important, we need to develop closer link with clinicians. And I'm very happy that I was told that there are several clinicians, medical students in this auditorium. This is key that we get closer link with you. We have to develop, for example, MD, PhD programs. So that means clinical scientists, clinician scientists that can really bridge the world of science and the clinical world. Develop biobanks, absolutely key, brain banks, clinical data. But for example, there are great things. We were told yesterday in Riyadh, for example, in the hospitals where you have those patients accessible, the data. So we need to share this data to try to better use this fantastic treasure, which is human genetics. I always say, the oil of the 21st century is data. Yes, data is the oil of the 21st century, and I think this country has a lot to offer. We need to develop more interaction with companies interested in neuroscience. So let me just give you one example. We're in Lausanne and very close in Vevey, about 10 kilometers from Lausanne, is the headquarters of a very large company called Nestlé, which is a nutrition company, probably well known from all of you. We've convinced them to create a new institute whose mission is to understand health and disease as influenced by genetics, metabolism, and environment, with the goal of translating this knowledge into what I call personalized science-based nutrition. So I would say prevention. So personalized prevention, and I think that the food based on your genetic proteomic data is a very interesting target. At EPFL, we promote the new startups. We create about 15 to 20 new companies per year on our campus. Those are just a couple of examples of startups in neuroscience. Aleva Neurotherapeutics, that does neural prosthesis, smaller electrodes, less invasive. AC Immune, an antibody that produces an antibody against Alzheimer's disease, tested by Roche, the big Swiss pharma. Amazentis, the company that I started with a couple of friends, that looks at functional food, brain health, and prevention. So you could see that those companies come from different fields: electrical engineering, computer science, food science, pharma, and I think this is very encouraging to see that this is today happening. The Center for Neural Prosthesis that we've created at EPFL, where we have already 5 and tomorrow 10 new faculty working on this real approach. And then we were very fortunate that Dr. Viss made us a fantastic gift of 100 million Swiss francs to create a new center for neuroengineering. So what we will do is to try to bring people from different backgrounds in a one and single area. So I really hope that this conference is only the beginning. You will see now a lot of illustration of what I give you from, I would say, a helicopter view. And I hope this will convince you that we're living a very unique time in neuroscience. So even though you could say the last decades were not that great in terms of bringing new therapies, I think with this kind of revolution of IT technologies and nanotechnology and cognitive science, we will see major breakthroughs in the years to come, and we'll be very happy to do this with this part of the world. So I will finish by putting this quote that I really love from Rudy Dornbusch, who's a professor at MIT. He said something that I thought was very interesting. He said, things take longer to happen than you think they will. And suddenly they happen faster than you thought they could. So I think you have to be there at the inflection point, and I think we are at the inflection point for neuroscience. Thank you for your attention.